today, Dr. Amy Williams from 23andMe. Amy has a background in mathematics and computer science. She double majored in those two in college. And then she did her PhD in computer science at MIT. After her PhD, she did a postdoc at Harvard Medical School with Debbie Wright and David Ashler. And then she also have a very short postdoc with Molly Petrowski at Columbia University. And in 2014, she joined Cornell as an assistant professor. And that's where I think my postdoc, I met Amy over there. And Amy was promoted and got tenure at Cornell. In 2022, she decided that she would left Cornell and joined 23andMe. And Amy's research has been focusing on phage building pedigrees. She's working on statistical genetics and doing method development, tool development for haplotype phage and building pedigrees. And one thing I admire Amy is that she's still doing a lot of the code writing, method development by herself. But let Amy share her research more.
Thank you so much, Kaishang. It's a real honor to be here, sitting with you all, and I'm looking forward to this. And please feel free to jump in and ask questions. I'd like this to be interactive. So we've entered an era in human genetics where we have sample sizes of unprecedented scale. So there's the UK Biobank with roughly 500,000 individuals. And then there's genetic testing companies such as 23andMe and Ancestry and others that have tested more than 20 million individuals. And this is very exciting, right? So we have the potential to harness these data and use them to build methods that have extremely high accuracy. So as an example, this is a plot from Brian Browning. This is so I'm going to be talking a lot today about phasing, and I'll define that in a moment. But what we have here is on the y-axis is a measure of error rate, and on the x-axis is sample size. And as you can see, the state-of-the-art phasing methods get more and more accurate as sample sizes increase. And there's the potential to leverage 23andMe's giant customer base to understand just what the limits are on phasing accuracy. Can we get this down to zero? So today I'm going to be talking about two problems. So I alluded to the first on the previous slide. So this first problem I'm going to talk about is what's called phasing or inferring haplotypes. We all inherit 2 copies of each chromosome, 1 from each parent. And so the physical reality is that at sites that vary, at polymorphic sites, we have site on each chromosome, and they co-segregate from each of our parents. Despite this physical reality of alleles being on one haplotype or the other, the most cost-effective ways to generate genetic data erase haplotype information. They erase the phase. So I'm sort of showing that here pictorially with a sort of purple chromosome where we have, we know physically along the length of the chromosome where the polymorphism live, but we don't know which allele is on the same haplotype, so T, or if it's on the same chromosome as T. So this is a problem, and it's a very important problem. And researchers have developed statistical ways of inferring haplotypes, but they do so imperfectly. So you can take these data and start inferring and get haplotypes where there are errors. So we've jumbled together sections that are perfectly well phased with errors that lead to switches in which haplotype is on the same chromosome. So these are called phase switch errors. These dashed lines show here. And so, you know, again, we're going to see what we can do to reduce switch errors. So going back to this exciting time of these very large data sets, in addition to improving accuracy for phasing, large data sets have many relatives. So you can imagine if we had a data set with 1,000 people, we compared you to those 1,000 people, the odds, if they were a random collection of people, the odds that you have a close relative in that data set would be quite low. But if we compared it to 10, 20 million people, the odds would be considerably higher. And because you're comparing to, it would be considerably higher. So in these large data sets, we have the potential to sort of compare all pairs of individuals and try to identify relatives and even build up pedigrees. And this is an important problem. Pedigrees are very useful in genetics. And something we can also do is use these relatives to reconstruct the DNA of the ancestors of those relatives. So throughout this talk, I'm going to show these pedigree plots. Individuals that are shaded will be those that we have DNA for, and unshaded individuals will be those that we do not have DNA for. And so because a parent in every generation transmits half their DNA to each of their children, we can use DNA from multiple descendants of a person to try to reconstruct their partial genome. So in this example, if we knew that this was the pedigree structure, we could compare this person to his first cousins and see that this red section is shared with one of those first cousins, and then infer that must have come from his mother that's also related to these individuals. And likewise, that this brother of that person carried that section of red DNA. So using these shared segments that are, the technical term for them is identity by descent segments. These are identical sequences co-inherited by two or more people from a common ancestor. And we'll show this pictorially as a color. Unfortunately, the data doesn't have the color in it. We have to try and figure that out on our own. So in this talk, I'm going to, the second problem I'm going to talk about is reconstructing the DNA from parents from a set of siblings. So it turns out a set of siblings inherits a lot of DNA from their parents. So the expected fraction increases exponentially. So it's this equation 1 minus 1/2 to the power s, where there are s siblings. And I've shown a table here with the expected proportions that you should inherit given whatever number of siblings. And as you can see, this number goes up very rapidly. Unfortunately, not all families have 8 children, but even with only 3 children, we can get on average 87, 88% of the parent's DNA. So why do we care? Well, we could care for several reasons, but we can use the data from those parents in genome-wide association studies. So we might have the complete health histories of these individuals and add them into our GWAS. So that will improve power in those genome-wide association studies. There's also a concept that I'm not going to get into much today or even at all, but there's a concept of genetic nurture where the untransmitted alleles of a parent, so your parent transmitted half of their DNA to you, the untransmitted alleles that they carry are actually associated with the phenotypes of their offspring. And the analyses of genetic nurture require parent data. So if you can reconstruct that data without the parents themselves, you can expand the scope of the number of individuals that we can include in these studies. And you can also use these parents' DNA to do more reliable relatedness inference. And the list could go on. So with that as a background, I'm going to talk about 3 things today. The first is to talk about, in some sense, a benchmarking study of phasing with state-of-the-art methods in massive data sets. And then I'm going to move on and talk about new methods that we've developed for interchromosomal phasing. And I'll define that when we get there. And then I'm going to talk about this reconstructing parents' genotypes from siblings. Okay, so first I'll go into this phasing millions of samples. So I don't I am now at 23andMe, and Jared O'Connell is my colleague there, my peer there, and he led some work where he was doing an analysis of the accuracy of ShapeIt and then Beagle on our massive data set. So Jared, and actually I should have put a photo, it's going to come up later, of Cole Williams at Brown University. So the two of them did phasing experiments. So the first done by Jared at 23andMe, we phased 6,000 individuals with a panel of more than 8.6 million research consented participants in the 23andMe data set. And there are roughly 1,000 from each of 6 populations that I will mention later. And then Cole Williams took about 1,000 UK Biobank trio children and phased them with together with 486,000 UK Biobank participants, and also included 1,000 Genomes Project individuals to get more diverse representation, because the UK Biobank is very European centric. So these are all trio children, and I'll get into why on the very next slide, but I should emphasize that the parents were not included in any of these analyses. So it should represent the phasing accuracy if you don't have the parents in the phasing method. So we're phasing chromosome 2, and the performance metrics that we're using are switch error rate. So that's just a measure of the number of switches that we saw, switch errors, divided by the number of heterozygous sites. And we used trio parents to phase the children in order to get the true set to compare to, true phase. And I'll define that in the next slide. And then we're also interested in the total runtime of these methods. Okay, so how do we get the truth? How do we know what the true phase is? Well, phase is defined as the set of alleles that each parent transmitted to you. If you have the parent's data, you can in principle just completely perfectly infer the true phase. So if dad here has a C and a T genotype and mom is CC, then if the child is CT, we can know that the T was transmitted by dad and the C by mom. So that works extremely well at almost every position. However, if the parents are both heterozygous, or if there's cases of missing data, there can be ambiguities, and we just ignore those for the purposes of measuring the accuracy of these tools, right? So, so that's how we get our truth set. Okay, so here are the results. So we've run, again, 6,000 real children in the 23andMe study and about 1,000 UK Biobank participants. So on the left panel, we have is a set of 6 populations: Europeans, Ashkenazi Jews, Latines, South Asians, East Asians, and Africans. And we've got on the y-axis is a measure in % of switch error rates. So the left side is 23andMe, and the right side, done independently, is UK Biobank with these individuals of 1,000 UK Biobank participants, together with some of these same populations, but from the 1,000 Genomes Project data. One thing you can immediately see is that the phasing accuracy is much, much lower in the 23andMe study than in the UK Biobank study, but quite good for the UK Biobank participants. And that there's variation in both data sets across population groups. This is largely driven by not only sample size, but population representation. So in the 23andMe, so again, the left is 23andMe data, this 8.6 million individuals. On the right is this roughly 500,000 UK Biobank. Unfortunately, both data sets are very Eurocentric, but there's a lower proportion of Europeans in the 23andMe study and a higher proportion of Latina and African American individuals, with some representation of East and South Asian individuals in the 23andMe data set. And this sort of correlates in a meaningful way with these results. So I'm going to get hard numbers in a slide or two, but the European, Latina, and African descent individuals, along with Ashkenazis, which are European population, have the lowest switch error rates. Now, one thing to note though is that the representation of Latinas and African Americans isn't anywhere near that of Europeans, but because of admixture, there's a meaningful part of their DNA that is European, and so the European component of the data set is enabling a better accuracy than in groups that are unadmixed and not as well represented. And so, you know, one takeaway from this study, which, you know, I don't need to tell the world this or tell you this, you know, we need to get better representation of individuals from many different population groups to improve the accuracy. And that's very much highlighted in this UK Biobank study that's so heavily biased towards accuracy in the European descent individuals than in others. I promised to get you hard numbers. So let's ignore the sort of right side of this plot for a second, just focus on this column. So in the 23andMe data set, the median raw switch error rates are very, very low. So again, these are percentages. So you could add sort of 2 zeros in front of the decimal to get the overall switch error rate. So this is a tiny, tiny switch error rate across many of these populations, and indeed all of them are less than 1%. But we can go a little bit further. So one of the most common forms of errors is what some have called a blip error. So imagine you're phasing a chromosome and there's a single site that has a switched phase relative to the truth. That would count as 2 switch errors, one in front and one sort of on the other side.
Unknown caller.
So this is sort of very harsh metric to use for this kind of, that's okay, this kind of error. So so what we did, and moreover, so if you have a single site that has an error, the odds that it affects downstream analyses is somewhat low. So we deleted these blips, these single site errors, and in fact, the median error rate in these more represented populations drops to 0, and the other populations drop by about half or so. We delete these blip errors. So that this is quite striking. We were excited to see that the median person in our data set was actually getting 0 switch errors, and that almost everyone was getting extremely low switch errors. So this is very practical for many downstream applications. So this is showing that same these error rates in terms of numbers of switch errors. So in black, you have the number of individuals from each of these populations that have 0 switch errors after you move the blips. And as you can see, Europeans, Latinas, almost Ashkenazis, and Africans have more than 50% of their samples with 0 switch errors. Again, unfortunately, the East Asian and South Asian individuals have a much smaller fraction with a perfect phase. And again, we would love and are seeking to increase our representation across all population groups for that reason. This left panel is much better in terms of the number of very low switch error rates in 23andMe versus UK Biobank, again, driven by the larger sample size in that group. Now just briefly, again, this is sort of a benchmarking study. We looked at the switch error rates of Shape It on the y-axis and Beagle on the x-axis in the scatter plot and compared them to each other. So I'll focus here on this subpanel. So this is a zoomed in plot of just the UK Biobank individuals, and what you can see is actually these methods are sort of at parity. They're getting roughly the same switch error rate in the UK Biobank, and notably, both these methods have been built with UK Biobank as one of their target studies, and they both do, again, at about parity. But if you look closely, you'll see that most of these points are above this dashed line, or many of these points, and so basically, Shape It has a higher switch error rate than Beagle does. And that's something just to note, and kind of interesting. And you wonder, I don't think this was intentional by any means, but you wonder how it came about that outside of UK Biobank there is this skew in accuracy. This remains true in East and South Asian samples where there's a higher switch error rate in Shape It and lower in Beagle versus in these individuals. Okay, so last slide on this topic, runtime. So if Beagle is more accurate, is there a reason for that? And presumably part of the reason for that is that Beagle takes longer. It's about 51% faster to run Shape It than Beagle in the 23andMe data set, and about 36% faster in the UK Biobank. So anyway, so this is exciting. We're interested as a company in improving the quality of phase, and I think academics as well are interested in. And the time will come, hopefully not too far off, when data sets on this scale will be available in academic labs. Also kind of interesting as far as pragmatics go, the compute cost for these 6,000 samples using this giant panel of individuals is only less than 1 cent per sample to do this. So it's very practical to apply. These runtimes sound large, but when you use big compute clusters, it's still very practical. So questions on this? Yes, Kaisha.
For the phasing, can you do it one individual at a time so that you have one new customer you phase it?
You could. If you study the compute time, it's there's a lot of overhead in reading in this massive data set. So the compute cost for one sample alone would be much higher. So there's sort of like this first phase is just read in the data and process it, and then you do the actual phasing. And so you don't sort of amortize that compute cost over all samples when you only do one at a time. Does that make sense?
Yeah, that's the sample size, the sample input, one sample or 1,000 sample. Is it going to impact the phasing accuracy?
So I don't want to say too strongly, but I believe that at least one of these methods, if you use a giant panel, it does not incorporate the input target individuals with the phasing. Yeah, so that's relevant to reproducibility, right? Yes.
Just as a sanity check, we're working with SNPs rather than like full genome.
Yes, this is SNP array data. So our data set is roughly 600,000 SNPs. UK Biobank is on that order as well.
I can ask another question.
Yeah.
If we want to, for the East Asian, South Asian population, if we want to reach the same very low switch error rate as Europeans, what sample size do we need? An absolute number for that?
I shouldn't speculate. A slide I didn't include, a plot I didn't include. So there's this interesting thing you can do, which is analyze the IBD coverage on the x-axis and the y-axis is switch error rate. What you can see is actually population membership is more or less irrelevant. It's all about how many relatives you've got in terms of your phase accuracy. So does that make sense? So so there are individuals that we're getting perfect phase for, and these are presumably people that we have just by happenstance, or maybe by their efforts. Does that make sense?
So it mainly come from the number of relatives in the
from IBD segments, right? Right. So how many people have these extended haplotypes that they share with the target individual? If you have good coverage for that, then you can do well with phasing. Okay, so now I want to talk about some methods development. So I promised that I would define interchromosomal phasing. So we can get effectively perfect phase for the median individual in our data set. What's interesting though is that actually misses quite a bit of information. So if I wanted to know, and I'll motivate why on the next slide, but if I wanted to know not just the phase of a person, but which haplotypes were co-transmitted by the same parent across chromosome 1, 2, etc., then I need interchromosomal phase. I need, in some sense, if you like this color analogy, not to get 44 colors or 46 colors, but to actually assign 2 colors to every single chromosome and implicitly sort of say, okay, all the red chromosomes were transmitted by one parent, all the blue by the other. We have to be careful. Unless we have a male, we have IBD sharing to the X chromosome, we may not be able to get sex labels. But we can get sex labels if we have male and we have IBD sharing to the X chromosome. Great. So there's this computational problem. Let's infer these interchromosomal phase, these colors across chromosomes that correspond to the same parent emitting each chromosome. Why would we do this? One of the reasons we would like to do this is that we can do parent of origin association testing. So there are variants that are associated with phenotypes only when they're transmitted by your mother or by your father. And this is driven by imprinting and there's been meaningfully sized studies of this, but there's more to be done. There's more information to be gleaned, and it's understudied because we lack data for parents. And what I'm showing you is that we can actually subdivide the genome, get, in some sense, parent of origin in males without the parents' data. You can also, and this is like something that's more company centric, but you can also subdivide a person's relatives into 2 groups. I could tell you, here's a set of relatives you share through one of your parents, parent A. Here's a set through parent B, and maybe get the sex of that parent in some cases. And customers are interested in this. Consumers are interested in this. Okay, so this is work that was led by Cole Williams. So I'm collaborating with Sohini Ramachandran at Brown University and her student Cole Williams. How are we going to do this, right? So like you might think for a second, like what is the signal that we could use to get linkage between chromosome 1 and chromosome 10? So basically relatedness, right? If I had a maternal first cousin, and these segments they share with me should all come through my mom, right? And so if we could subdivide the relatives into 2 groups, and sort of the reverse of the last slide, if we could figure out which relatives are on each side, then we can get a lot of signal. So if we have a maternal first cousin and these orange segments are shared with that first cousin, then we could use that to sort of do this interchromosomal phasing and fix up errors in that way. One thing to note that I'm not going to get into, but it is a confounder in some cases, is that not all our relatives are shared through only one parent, right? And actually this can be a meaningful issue, but I won't get into it too much. Okay, and then so that's one signal. Use relatives, right? Use relatives to sort of phase these different chromosomes. The other signal, or another signal that we have, is when 2 segments are on opposite haplotypes, 2 IBD segments. So this orange and green segment must have been transmitted by distinct parents, right? So we can use this signal for phasing. Okay, so how does the algorithm work? So the way this works is that we take these IBD segments, so these colored red and blue segments are IBD segments, and they're laid out along the length of the chromosome. And we can build up what we call tiles, which are a set of IBD segments that overlap each other irrespective of haplotypes. So you can imagine, so note that this segment shared, so these labels are for the relative, so an identifier for the relative. So segment D and E1 do not overlap each other, but they both overlap segment that is shared with B. And so they are part of the same tile, right? Okay, so we build up tiles. So there would be a tile on this left side and a tile on the right side. The next step is to build a signed graph that links these IBD segments. So this may look complicated, but it's fairly straightforward. So we are going to draw edges between these IBD segments. We will draw a positively weighted edge between segments that are on the same haplotype. Those should have been inherited from the same parent. And we will draw positively weighted edges between segments transmitted by the same relative, in this case, relative E. Because we believe that in most cases, those should have been inherited from the same parent. And now, kind of a twist, we're going to draw negative edges. We're going to try and force away segments that are that are inferred to exist on different haplotypes. Right, so we draw together the first two signals and push apart the other signals. Okay, so we build the signed graph, and then we apply spectral clustering. I'm not going to get into the details on this, but you can convert this signed graph into a signed Laplacian matrix and apply spectral clustering. And often this actually fairly cleanly divides these segments into two groups. That doesn't mean there aren't mistakes, but you get individuals, relatives assigned into exactly two groups. Okay, so that's the algorithm. So the metrics that we use, the metric that we use for assessing phase quality is different. We're no longer going to calculate switch errors. We're going to be even harsher now. Instead of calculating switch errors, we're going to calculate the Hamming distance. So if I looked in this region here, there are just these two switch errors. But if there were 10 or 100 SNPs inside this region, that would count as 100 errors in our analysis. So we're going to be even harsher now than we were before. Calculate the Hamming distance between, so we have two possibilities. It might be that haplotype 1 is maternal or haplotype 2 is maternal. So we're going to calculate the Hamming distance of both possibilities and take the minimum of that. We're not right now, and actually not for this talk, going to try to infer the sex of the parent, right? So either possibility applies, and we will compute the statistic depending on context, both intrachromosomally and also interchromosomally when we're concerned with genome-wide phase. Okay, so now results. So the first question is, are these tiles well-phased, right? So is it right to sort of pull together all these IBD segments? Do you actually get, right? So there could in principle be a switch error within a tile, right? You'd hope that would be rare, but there could be. But what we have here, and I'm not going to go through this, there's a lot of information here, but what we have here on the y-axis is this within tile intrachromosomal phase error on the y-axis, and then the x-axis is a measure of the number of heterozygous SNPs in the tiles. So presumably longer tiles have lower switch errors, and in fact, you know, if you want to squint, it's not super important, but there's some relationship there. So overall, though, there's a very low error rate in these tiles, switch phasing error rate. Okay, so now the main set of results. So now we're going to look interchromosomally. So I've got, you know, let's say, you know, 10 tiles for straight of those 10 tiles, right? Let's throw out the rest of the genome, because in some sense, we don't have a lot of information, right? But just throw out the rest of the genome. How are we doing? So again, these are percentages. And again, this very harsh statistics. In the UK Biobank, we can get 0.09% phasing error rates within these tiles, even lower lower in 23andMe Europeans. So the leftmost plot here is UK Biobank. The rest are 23andMe samples. So in Europeans, Latinos, and Africans, those same populations that were doing well before, we're doing quite well. I'm sorry to say that in other population groups, we're not doing nearly as well. And presumably this is due to similar reasons. It's the metric's much harsher, but to similar reasons that we need more relatives from these groups to do better. So that's within tiles. Now we want to do Kaisha.
One thing I noticed is that for the Ashkenazi Jews, before it has very low switch error rate, but in this case, it's the highest.
Yes.
What
Yeah.
happened to that group?
Yeah, so the bottom, I mean, unfortunately endogamy, so Ashkenazi Jewish people, there's high levels of relatedness. And so most Ashkenazi Jewish people, this has been shown in other papers, are related to other Ashkenazi Jews at like the fourth cousin level. And so there's confounding between, you find a relative, it's not just a maternal or a paternal relative, it's both. And that's confounding us here. And it'd be interesting to think about strategies for improving this. I do think sample size and getting people that are truly much more closely related is a big part of the answer there. Okay, so now let's start extending this out to the rest of the genome. We looked in tiles. Now, what about a chromosome that has a tile on it? We do well there. The answer is yes. So in several of these populations, so these are all median phasing error rates. So unfortunately, UK Biobank, the sample size isn't sufficient to continue to do extraordinarily well. But in 23andMe, in at least these population groups with many relatives, Europeans, Latinos, and Africans do quite well, less than, or about 2% phasing error rate. Okay, so last sort of metric, what about the rest of the genome? So so you basically have a random guess. If you have no IBD segments, no tiles on a chromosome, you're just going to be completely guessing about what the true phase is. And so basically you should expect these numbers to go up, and they do, but not by a lot. So actually, I think I copy pasted some of these numbers wrong, but I apologize. I can dig this out, but anyway, the numbers are about, again, 2%. It doesn't get that much work. And basically the reason for this is that most chromosomes in these populations have an IBD segment. The number of relatives is quite high. But I don't want to sweep under the rug the fact that several population groups are still doing somewhat poorly. Now, Ancestry DNA somewhat scooped us. They released a paper, I think it was about a year ago, on the same topic, and their error rates in West Europeans and in Latinos there's a mix in the populations that we're doing well in. We seem to do about the same, or maybe slightly better or worse. But in some of the populations, so South Asians, they're doing somewhat better, although I don't think 26% is anything to be too excited about. They have a bigger sample size than us, so they are able to do somewhat better. And there may be some sort of phase shift where you get above a certain tipping point in terms of sample size, and then the phasing error drops quite dramatically. Maybe something we can look into in the next little while. Okay, so any questions on this before I last topic? Okay, so lastly, now switching gears somewhat, we want to try and reconstruct DNA from individuals, from the ancestors of individuals. So this might seem kind of simple, right? We've all got 50% DNA shared with our siblings roughly, and find these shared segments. But it's actually complicated, and I'll get into why more later. But what one way of thinking about this is, if you are familiar with the term, it's inferring a one-generation ancestral recombination graph. We're going to trace and identify the recombinations and so on. And I'm not going to get into many details on this, but the first thing we do is we infer phase with a method that is designed for nuclear families. And this will work well, but there are ambiguities. I'm going to talk about this in more detail in a moment. There are ambiguities for smaller families, but with bigger families, we can do quite well. So we infer that phase, and then we're going to resolve the ambiguities using IBD segments to other relatives and sex-specific maps. So I said a lot. I'm going to now dig into all this in the next several slides. So, okay, so how do we do phasing of families? So if we were marching along, we're doing this, right? And we knew that the children had inherited these sort of, first child gets a blue, second child gets a pink, next child gets a dark blue and a red, and so on. If that's the inheritance pattern up to a given marker, now our question is, we know what the genotypes of these children are, what's the genotypes of the parents, right? So I'm going to tell you that, I mean, there's a lot to say here, but just, I'll tell you that one of the parents is heterozygous and one is homozygous, and you can get that from this, right? Or you can roughly get that from this. So distilling a lot of details down, there's a couple phase possibilities. One is that the mother has the T allele and the father is homozygous for C. If that were true, then this is one possible way of assigning the alleles to haplotypes, and there would be 0 recombinations in that case. Now, what if instead the dad carried the T allele, right? If that were the case, then we could assign this T allele to the dark blue chromosome. But as soon as you do that, every other chromosome is C, and you have induced recombination events, right? So this second child and the third child have inherited a crossover relative to the previous site. And this is possible, but it's much less likely, right? We all know recombinations happen, but at any one locus, the rate of recombination is quite low. So it's much more likely that the true phase is this first possibility. And that we can assign the alleles to the parents accordingly. Okay. Now, there's a lot of details here, and it's not crazy complicated, but we don't have a lot of time and it's a little mind-numbing. So I'll just tell you that Happy implements a hidden Markov model, and the parents' haplotypes are what is constitutes, and the part of the hidden state is the parents' haplotypes. Okay, so I mentioned ambiguities. So it turns out that there are cases, such as this one, where you can, so if the parents, one parent is CT, the other is CC, so you can assign the haplotypes in either orientation, right? So if T is on the pink chromosome transmitted by mom, or if T is on light blue chromosome transmitted by dad, both of those lead to 0 recombinations. So from the data alone, we can't actually, from these children alone, we can't tell whether dad is homozygous and mom is heterozygous or the reverse. That's annoying, but something we just have to live with. And again, this leads to ambiguities. Okay, so we run Happy. So Happy at many positions is going to tell us the genotypes and in fact the haplotypes of the two parents. And so we're going to evaluate Happy, and I'm going to talk more about the IBD sharing in a moment, on data from the San Antonio Mexican American Family Studies, where there's these giant pedigrees. So this isn't even the biggest one. This is one of the bigger ones, but it's not even the biggest. There's 114 nuclear families where we have data for the parents. We want to know what the truth is, and 3 or more children. So we use those data, and then we use 23andMe research participants, where we randomly sampled 220 families with between 4 and 10 children. So the first thing we're going to do is actually much easier than what I just talked about. We're going to include data for one of the parents. So we will censor either the dad or the mom, run happy and only happy, no IVD segments to relatives, and we'll see how much of the data, the parents' data, we can reconstruct. So what do we get? We're reconstructing only one parent. We can reconstruct between 78 and effectively 100% of the parent's DNA, which is really exciting. The amount you can reconstruct depends on the family size. So for families with between 3 and 4 children, the range is between 78 and 96%. And with 7 children or more, it's up to, again, 100% and more than 96%. That's pretty exciting, right? And actually, kind of fun, I've got a website, happydna.com, where you too can reconstruct, and I use it to reconstruct my grandmother's DNA. I genotyped my grandfather, but my grandmother had passed away before this was widely available, so I'm kind of excited. I'm a consumer and excited to get this. Okay, so actually, very importantly, we should be concerned with what our error rate is, right? So anyone can tell you that they have reconstructed your ancestor's genome, right? I can use a random number generator like anybody, right? But what we really want to know is it reliable? Is this true? And in fact, the error rate is less than 10⁻⁴ per site, which is comparable to, maybe even better than a SNP array genotyping error rate. Okay, now harder problem. Can we reconstruct DNA from both parents? Now I've changed the colors here. We've got green and gray because we can't actually tell which belongs to which parent. So what can we do? This is actually, I mean, if you think about it for a second, this is a very big problem. It's like an interchromosomal phasing problem. But it's even worse. The ambiguities cause a single chromosome to kind of break apart into independent regions. So we're going to use, just like interchromosomal phasing work, we're going to use IVD sharing to relatives. And hopefully, each of these relatives is related through only one parent, right? And if that's the case, then we can assign these segments back, and then we can once again infer the sexes of the parents using the X chromosome in male siblings, if there are any. But there's one more signal, so it's kind of exciting. Recombination in men and women is quite similar. The biological machinery is quite similar. But there are differences, and in fact, they're quite marked differences. What I have plotted here is this chromosome, human chromosome 10. On the x-axis is physical positions, the chromosome sign up on its side, and then the y-axis is crossover rate. And what you can see is that male, sorry, this is a little small, blue is male, red is female. Males tend to transmit almost all their crossovers near the telomeres. It's not zero elsewhere, but it's mostly in the telomeres, whereas female, I wouldn't call that uniform, but it's much more spread out throughout the chromosome. So we have used this in the past. There's a method developed by a student called student Ying Chao and Jen Sanarud called CREST that uses this signal in half siblings, only autosomal data, to infer the sex of their shared parent. So this is real data here. The truth is either pink or blue, where for the true sex, and this is able to separate these very well. And in fact, it's not a giant data set, but we got it completely correct in those data. So we're really excited about that. Our method, Happy Recap, also incorporates this. Okay, so we now, now getting to the results. So bring you back. So we've got a set of siblings reconstructing both parents' data. How much can we reconstruct? So when we looked at all families, this number is kind of scary. Like some of these people didn't have close relatives. So you get tiny, tiny fractions, and that fraction is only driven by this recombination brace signal. So in fact, the range is more like 30 to 96% if you require a relative. The mean sharing is about 74%. And the median is 77. So it's actually median statistics are a little better here. So that's using all families. When you use families with small numbers, 3 or 4 children, you can reconstruct up to 83%, mean of 62%, its median 70. And when you use these very large families, 7 or more children, we can reconstruct up to 97% of the parent's DNA. You might wonder, why is this number not closer to 100%? And the answer is that there's other forms of ambiguities. If the children are entirely heterozygous, there's actually no signal, there's very little signal there about which allele came from which parent. And so the mean reconstruction rate is about 90% in these large families. And excitingly, the error rate is very, very low. So then summarizing, you can reconstruct very, very accurate parent genotypes with this using family-based phasing, IVD sharing, and HAPI just by itself can get you large fractions of the parent's genomes. So in conclusion, I hope you are convinced that you can get extremely high accuracy phasing when you have, the double-edged sword of this is it's like both tantalizingly exciting and very disappointing in certain populations. But the great news is there's we can all spit, right? And actually, I don't mean to push this company. We can all get tested in various ways and collect bigger data sets and improve genetics for the entire world. So we can do phasing when we get huge data sets at unprecedented accuracy, and we can do reconstructions of parents, large fractions of parents' genomes at basically the same accuracy or even better than if you had run a genotyping array on that person. So there's a lot of people to thank. So first, customers and individuals that tested with the San Antonio Mexican American Family Studies made this work possible. Jared O'Connell's work on phasing in our giant data set sort of spearheaded our move towards not developing a new method, but using existing methods. Why not? Cole Williams led the interchromosomal phasing and all the UK Biobank analyses of the first topic, and Chris Canu and Sohini Ramachandran are co-PIs on that work, and Ying Chao led the work on HAPI recap. And I want to thank my colleagues on the Population Genetics Research Development team at 23andMe and the investigators with San Antonio Mexican American Family Studies, especially John Langero. Thank you for listening.
Thanks. First of all, I want to apologize for my phone.
It happens.
Anyway, I'm wondering, how can you detect or infer or adjust for chromosome rearrangements in individuals, the hidden ones, obviously, that either, I'm thinking versions, translocations, even copy number variants, duplications, deletions, and that's going to complicate the phasing, right? And
Yeah, so inversions, I would suspect shouldn't be a terrible problem for phasing, because within the inversion, there's very little recombination, and actually, sort of, if anybody else in the sample has it, you should just basically get it.
Yeah.
There would be issues at the edges, but hopefully those would be minor.
but in terms of the tiling,
So the tiling, yeah, it's an interesting question. Sure. Yeah. It's funny. I haven't thought much about it. I think I want to say it's not terrible because again, thinking about it, version and deletions might be an issue, but I kind of want to come back to your question, like let it percolate you for a moment. But I think it should be okay. Let me think about it.
So we don't have, basically we don't have time, we're running out of time.
Yeah.
So if you have questions, there will be lunch with Amy. So bioinformatics students to stop by, ask questions, and thank you again, Amy.
Sorry.